Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. If you're finding value in these deep dives, please take a moment right now to like this video and subscribe to our channel. It truly helps support the Science Experience and allows us to keep bringing you these fascinating scientific explorations. Today, we're taking a deep dive into a groundbreaking paper from Cell, just published October 2, 2025. It's by Zhu et al. From researchers over at Caltech and the Weizmann Institute of Science. And this team has, well, they've really shaken things up, revealing new principles of co-translational mitochondrial protein import. Now, just to set the stage for you, think of mitochondria, right? They're the ultimate powerhouses of your cells, absolutely crucial for, well, everything from generating ATP, that's your body's energy currency, to regulating calcium. And for them to do their job properly, thousands of different proteins need to find their precise way inside these organelles.
Exactly. And for decades, the sort of fundamental puzzle for cell biologists has been this. Almost all those mitochondrial proteins are actually synthesized out in the main cellular space, the cytosol. So the big question has always been, how do these proteins find their way from that really crowded, chaotic environment into the very specific, highly organized mitochondrial compartments? The established view for a very long time heavily focused on what we call post-translational import, which basically means a protein is fully made, might fold up a bit, and then it's somehow guided to and imported into the mitochondria. It's kind of like mailing a fully assembled package, right?
Right, the finished product gets shipped. So that's the established wisdom. But, often the most exciting science comes from challenging those very assumptions. And this new research delivers a truly fascinating curveball. It really makes us rethink that whole classic view. It reveals that nearly 20%, 1/5, of human mitochondrial proteins are imported co-translationally. This isn't just some, minor detour. It means these proteins are actually being pulled into the mitochondria while they're still being made on the ribosome. It's like, I don't know, building a bridge as you're crossing the river.
Simultaneously.
Okay, let's really unpack how they even saw this happening. So this is a huge finding, but how on earth did they actually see this? Catching proteins literally mid-synthesis, heading into the mitochondria. What was the clever experimental trick they used?
That's where their really elegant technique comes in. It's called selective ribosome profiling, or SIR-ART. You can imagine it like literally snapping a molecular snapshot, if you will, of a ribosome that's still actively churning out a protein chain. But crucially, that protein chain is already interacting with the TOM complex.
The TOM complex, that's the main doorway, right? The translocase of the outer mitochondrial membrane.
Exactly, the main entry gate. So SIR-PS allowed them to precisely identify which ribosomes were physically paused right there at that mitochondrial gate mid-synthesis. An incredibly precise way to see these fleeting, really dynamic interactions that were, frankly, very hard to capture before.
And what they found wasn't just some, rare event.
Not at all, no. What's really fascinating here is that this con-translational pathway isn't just a minor side route, some kind of biological detour. Their data showed it's truly significant. It accounts for nearly 1/5 of the entire collection of proteins, the proteome within the mitochondria in human cells. That's a substantial portion. It suggests a much more widespread and fundamental role for this import mechanism than we ever really appreciated. It means a significant chunk of mitochondrial function actually relies on this pathway.
Okay, so we've established what this pathway is and just how widespread it is. Pretty significant. But the next logical question is, well, who is it for? Which proteins use this special route? For years, the thinking, especially from studies in yeast, suggested it was mostly about membrane proteins, those sticky hydrophobic bits that might clump up or misfold if they hung around too long in the watery cytosol. So the idea was they needed to be tucked into a membrane environment, ASAP. That kind of made intuitive sense. But this paper delivers its first major surprise here. Human cells seem to tell a different story.
Indeed, they do. They rigorously tested that exact hypothesis for human cells and found, well, membrane proteins were not preferentially enriched in this co-translational pathway.
Really? So not the membrane proteins?
Nope. Which naturally raises an important question. If it's not primarily for membrane proteins, then what kinds of proteins are prioritized for this co-translational import in human cells? And the data reveal a very clear pattern, almost counterintuitive at first glance. This pathway strongly prioritizes large mitochondrial proteins, specifically those over about 350 amino acids in length. And it's not just sheer size either. It's also proteins that have multiple domains and a really complex topology.
Right. They even have that clever metric for complexity you mentioned, the absolute contact order, or ACO scores.
Yes, the ACO scores. Think of it like a sort of blueprint complexity rating.
The higher the ACO score, the more twists, turns, and intricate folds the protein has. It means it's likely a real challenge for the cell to build and transport correctly. It indicates how many parts of the protein chain need to interact with other parts that are actually quite far away in the linear sequence, leading to a very convoluted structure.
I kind of like thinking about it like this. Imagine you're trying to build a really big, intricate Lego sculpture. If it's a small, simple one, right, you can build it completely outside a room and then easily carry it through the doorway. That's kind of like post-translational import. But if you're trying to build a massive, really complex Lego sculpture, well, it's far easier and more efficient to pass the pieces directly to the builder inside the room, assembling it bit by bit as they arrive. Instead of assembling it fully outside and then trying to squeeze this enormous thing through a tiny door. For these big, complex proteins, cotranslational import significantly enhances their import efficiency. It likely prevents them from misfolding or forming those unmanageable clumps aggregates in the cytosol. That would be a massive headache and a huge energy drain for the cell.
Exactly. Basically, the cell avoids a kind of molecular traffic jam that could bring everything to a grinding halt. It's quite smart, actually. And if we connect this to the bigger picture, it suggests a very clever, very nuanced cellular strategy. Cells seem to minimize their energy use by mostly importing proteins post-translationally. That's the default express lane, if you like. But they reserve this cotranslational route, this sort of special handling service, for those particularly challenging ones, the ones prone to problems. It's all about balancing cellular efficiency with really robust quality control, making sure these critical, complex components get where they need to go and in the correct shape.
Okay, this next part. This is the bit that genuinely blew me away. You'd think, wouldn't you, that as soon as a protein's address label, that mitochondrial targeting signal, the MTS, pops out of the ribosome, bam, it'd immediately be recognized and whisked away. Seems simple, right? Efficient. But the paper reveals something far more intricate, maybe the most surprising finding of all. That's not what happens.
Precisely. It's quite astonishing. The research shows there's a significant and, frankly, really unexpected delay. Import doesn't actually kick off until the nascent protein, the protein still being made, has grown to roughly 350 amino acids long, on average. And while that's the average, they saw a peak around 400 amino acids. But the range was quite broad, anywhere from maybe 200 and up to 800 amino acids. That's a substantial length of protein that's already been synthesized and is emerging from the ribosome before the import machinery fully engages. Even though that MTS, that address label, emerged much, much earlier in the synthesis process.
Whoa. So what dictates this delay then? It's clearly not just about time passing or some general minimum length. The crucial trigger they found, the thing that says, okay, now we go, is the emergence of a large globular domain from the ribosome. So basically a compact, often sort of spherical, folded region of the protein itself. This acts as a kind of second signal, doesn't it? It essentially disinhibits that N-terminal presequence, the MTS, and allows the actual targeting to start. It's like the address label is there all along, but you also need to see a specific type of package emerge before you actually send it off, like a two-factor authentication for protein delivery.
It truly is. That's a great way to put it. This multi-layered sorting mechanism implies that the N-terminal MTS is somehow shielded or inhibited early in translation, perhaps by interactions with the ribosome itself maybe, or possibly with chaperones out in the cytosol. We're still figuring out the exact details there. But then the emergence of that large, complex domain overcomes this inhibition. It likely does this by favoring internal protein interactions. Essentially, the protein starts to fold a bit on itself. And that folding, in turn, exposes the MTS properly so it can finally be recognized by the TOM complex at the mitochondrial surface. It's a really elegant sequential control system. It seems designed specifically to ensure these large, complex proteins get where they need to go on time and without causing cellular traffic jams of misfolded or aggregated proteins.
Amazing. And just to clear up another long-standing idea floating around in the field, the paper also found that the nascent polypeptide-associated complex, or NAC, something thought by some to be pretty key in mitochondrial targeting, turns out not to be required for this timely cotranslational import in human cells.
That's right. Another assumption, beautifully challenged and clarified by the data, it really highlights how much we're still learning about these fundamental cellular processes, doesn't it? And also, our human cells can often have distinct mechanisms compared to model organisms like yeast, where NEC might play a different role.
Okay, so wrapping this up, what does this all mean for you, thinking about the big picture? This deep dive into cotranslational mitochondrial protein import reveals a truly sophisticated, multi-layered protein sorting strategy. It's much more complex than we thought. The core insight here, I think, is that the cells have this incredibly intelligent sort of two-tiered system for protein delivery. There's a default express lane for most proteins, the post-translational route, and then there's a specialized premium service, the cotranslational route, for the really tricky ones, those massive complex structures that might otherwise gum up the works. This isn't just about efficiency, though. It's a brilliant quality control mechanism. It ensures our cellular powerhouses function flawlessly and helps maintain mitochondrial health. And as we said at the start, that's crucial for so many fundamental cellular processes, from energy production all the way to cellular signaling.
It really does make you wonder, doesn't it? Given this exquisite timing and signaling, this delay mechanism that requires a second structural signal to emerge, what other hidden delays or subtle molecular triggers might be at play in other cellular transport processes, things we haven't even discovered yet? There could be a whole world of these complex regulatory systems just waiting to be found, ensuring everything in our cells operates with truly astonishing precision. Something for you to mull over, perhaps, as you think about just how incredibly intricate our cells really are.
Absolutely. Well, that's it for this deep dive. We hope you enjoyed exploring the fascinating world of protein import with us. Remember to like this video and subscribe to the channel if you haven't already. It really does help us continue bringing you the latest science. Thank you for joining the SANS Science Experience.